Okay, so we are acidic chyme in the duodenum. Duodenum's about the first 1 foot of the small intestine. We've added in bile, we've added in pancreatic enzymes and bicarbonate. So we're really kicking into gear our carbohydrate, protein, and triglyceride digestion. As the material moves through the small intestine, it goes from the duodenum into the jejunum, which is about 3 feet long, then into the ileum, which is about 6 feet long. So during that whole time, we are doing most of our chemical digestion. We're also going to begin our absorption of the nutrients. So by the time this material gets to the end of the ileum, we're pretty much done with all the absorption and all the digestion. So the large intestine, which starts here in what's called the cecum, is going to begin removing more of the water so we can kind of compact this material. Notice the appendix is sticking off of the cecum. The appendix is often referred to as vermiform appendix. Vermiform means worm-shaped. Someone thought it looked like a little worm sticking off of that cecum. So the small intestine is very long, up to 10 feet long, but it's smaller based on its diameter compared to the large intestine. Notice we've got mesentery. This is all the mesentery holding those small intestines. They've been kind of moved off to the side here. Okay, let's look at the high surface area of the small intestine. So that's going to allow all that absorption to occur. The more surface area you have, the more you can absorb into your bloodstream or lymphatics. So the small intestine has basically 3 levels of folding. It has this macroscopic folding, these kind of circular folds that you could see with the naked eye. Then along that we have smaller folds known as villi, and then along the villi there's actually microvilli, all right, which are microscopic little finger-like projections. So the combination of these 3 largely expand the surface area of the small intestine for absorption. Now, the small intestine also produces some enzymes. Now, remember, up till this point, they were all coming from the pancreas. But the small intestine will produce enzymes we call brush border enzymes. They refer to brush border because the microvilli kind of look and make that small intestine look like a brush. And these enzymes kind of sit in those microvilli or along those microvilli and basically finish off the digestion. Once we finish off the digestion, we're left with monosaccharides and amino acids and smaller lipids, and those will get absorbed into the cells of the small intestine called absorptive cells, and then eventually they get absorbed into either the blood vessels in this villus or that lymphatic capillary called a lacteal. So the brush border enzymes are sucrase, lactase, and maltase. So those enzymes are going to digest disaccharides into monosaccharides. So the disaccharides that are digested are sucrase, sucrose. I even screwed up. If it's ACE, it's an enzyme. So sucrase digests sucrose into the 2 monosaccharides, glucose and fructose. Lactase digests lactose into individual monosaccharides. And then maltase digests maltose. So if you put an O here, it's a disaccharide. It's a sugar. If you replace that with the A, it's the enzyme that digests the disaccharide. So these will finish off your carbohydrate digestion. So as a result of their action, you have monosaccharides. You're going to have that lipase. It kind of functions as a brush border enzyme, but it's not produced by the small intestine, but it's going to be finishing up your triglyceride digestion. And then you have a number of peptidases. Peptidases are digesting the smaller proteins known as peptides, eventually down into individual amino acids. All right, so we do most of the absorption in the small intestine. We did most of the digestion there as well. This is a summary of our chemical digestion. So you can kind of spend a little more time on this slide than I will. But you'll notice I kind of color coordinated the enzymes. So if you're in blue, you're digesting carbohydrates. All right, you're either produced by salivary glands, the pancreas, or the small intestine. If you're in red, you're digesting protein. So your pepsin in the stomach, trypsin, chymotrypsin in the small intestine by way of pancreas, and then peptidases are brush border enzymes. And then if you're in green, you're digesting your triglycerides. So pancreatic lipase being the number one lipase. And don't forget, if you're in the small intestine, you're going to have bile that's helping out those lipases. So that's a summary of your chemical digestion, the enzyme, what they digest, where they function, who makes the enzyme. Absorption. So remember, the absorption differs based on whether you're a monosaccharide and amino acid versus a lipid. So what this shows, this cell is one of those absorptive cells of the small intestine. We see the microvilli here. If you're a monosaccharide like glucose, you're going to go into the bloodstream. And remember, that blood is going to eventually go to the liver by the hepatic portal. And then depending on how much sugar you have, you might store it as glycogen. The liver might convert it to triglycerides and send it out to adipose tissue. So it all depends on, the liver determining what to do with those monosaccharides. Amino acids, same kind of idea. You're going to absorb them into the bloodstream, send them to the liver. And remember, one thing that liver is going to do is remove the amino group. And then the liver decides, what am I going to do with these amino acids? All right. The dietary lipids. So remember, you're going to be digesting them with lipase. You're going to be using bile. You're going to absorb them in. You're going to often have to reform them into a triglyceride. So we kind of temporarily digest triglycerides. So you break them down into fatty acids and monoglycerides. You kind of surround them with some bile salts, which is called a micelle. You bring those in, you reform the triglycerides, and package them in chylomicrons. And remember, the chylomicrons are the lipoproteins that are going to transport these dietary lipids, so the lipids you just absorbed, into the lymph. And then that lymph is going to transport this to the subclavian veins. And that's how we get these dietary lipids into the bloodstream. Now, there are nutrients we never had to digest, so we're just going to absorb them. So those would be your vitamins and your minerals. And those are typically going to go into the blood unless you are a fat-soluble vitamin. So vitamins A, D, E, and K are going to go into your lymph. Okay, so that's absorption. What else do we have to do? Well, this is, I guess, more absorption. So this is just another image. Same information as the previous slide. But you'll notice here's one of these villi or a villus. These are all the nutrients we absorbed. I'm just showing you that your monosaccharides, your amino acids are going into the blood, going to the liver. So there's that hepatic portal. And then notice in here, these are your dietary lipids. They're going into the lymph. Eventually, they go into the thoracic duct, up that thoracic duct, and then into the left subclavian vein. So that's, again, absorbing those fats into the lacteals. All right, now we're going to be on to fluid balance, and then we'll talk about the large intestine and then we're pretty much done. So fluid. If you think about fluids that enter the GI tract, there's a lot more that enters it from organs than the amount you ingest. So you ingest, on average, about 2 liters of water a day. All right, so about 2 liters go in a day from your mouth. Now, there's a ton more fluid that enters from organs. So you'll notice on this figure, you've got about 1 liter of saliva is going to enter the GI tract a day. So that's a lot of spit you produce, all right, and swallow. About 2 liters of gastric secretions, about, a little over 1 liter of pancreatic secretion, almost 1 liter of bile, so 0.7 liters of bile, 2 liters of small intestinal secretions. So what this fluid does is helps deliver things in. Percent of it. Now that's going to leave about 1% of this water in feces. And that's what you want, because you don't want your feces completely dry, because that's going to cause, severe constipation. So you got to leave a little bit of water in that feces. All right. Now, what happened to that, other water? Well, you're going to urinate it out, right? A lot of this water gets absorbed in, and then you urinate it out. So if you ingest about 2 liters of fluid, you're going to be urinating out about, probably a little less than that. Okay. So that's fluid balance. Okay, now we're on to the large intestine. So a little bit of the anatomy first. So we're entering from the ileum. That was that last part of the small intestine. There's another little sphincter there, or in some cases, this one's known as a valve. It's the ileocecal valve because it's between the ileum of the small intestine and the cecum of the large intestine. This first kind of pouch area, it's usually a little wider than the rest of the large intestine, is known as the cecum. And in that cecum is where you find a lot of bacteria. All right, the bacteria love being in that cecum because if there's any nutrients that didn't quite get digested and absorbed, they're going to be entering right from that ileum. So a lot of bacteria reside in that cecum. There's the appendix sticking down from the cecum. So then the rest of the large intestine goes like this. You've got an ascending colon. Then there's a little bend here called the right colic flexure. There's the transverse colon because it transverses, it goes horizontally. Then there's another little flexure there, a little bend. Then you have the descending colon because the material now is going down. There's an area called the sigmoid colon. It's kind of an S shape. Notice here in the actual image, you can see that, S-shaped curve to it. And then the material, by that point, it's pretty much done. We've removed more water, so the feces has become a little more compacted, and it enters the rectum. And the rectum is where you can just, hold on to that feces until you find, a good location to deposit it. All right. You'll notice that the large intestine has a very distinct kind of pouch look to it. Those are called haustra or a haustrum. So we often refer to the movement as haustral churning. So it's moving the material from haustrum to haustrum. All right, all the way down until you get to the rectum. So that's the main anatomy of the large intestine. The functions, moving the material, haustral churning, using some peristalsis to move that by this point we would call it feces. It houses a lot of bacteria that are going to digest some of our nutrients that we didn't. The problem is bacteria digest molecules a little differently than we do. So they're going to produce gas as a byproduct. They're going to produce molecules that have an odor as a byproduct. But because they're living in there, they actually produce some vitamins for us. So they actually give off some vitamins, such as some of the B vitamins and vitamin K. So that's another, benefit of having bacteria. They take up space, so no pathogens can really take up space, but they'll also produce some vitamins for us. The large intestine is going to absorb water. In that water, there might be some ions and vitamins as well. And then ultimately, we form the feces. And then obviously, we got to get rid of that through the process called defecation. So no enzymes produced by your large intestine, absorption of water and electrolytes, compaction of feces, and then elimination of feces. How does that happen? We call this the defecation reflex. All right, so it's controlled by nervous system. All right, and part of this process is involuntary, part of it's voluntary. So notice there's the feces. All right, there's your poop. It's sitting in the rectum and it's stretching the rectum. All right, so the rectum knows when there's feces in there because it has stretch sensors. And those are on these green sensory neurons. All right, so when the feces enters, it stimulates the sensory neurons. They're going to head into the spinal cord and then cause a reflex right back out. So reflexes are involuntary. All right, and this is a parasympathetic neuron, and it's going to then lead to stimulation of the wall of the rectum. Okay, so the rectum's going to squeeze and start to push the feces. At the same time, we have motor neurons that are going to cause relaxation of the internal anal sphincter. So that's involuntary. Involuntary parasympathetic neurons contract the rectum, relax the internal anal sphincter, which is smooth muscle. So smooth muscle is involuntary, so you can't control that sphincter. But luckily, notice you've got a signal that can come down from your cerebral cortex. All right, so you typically will send a signal up. That doesn't show this, but you got to tell the brain this is happening. If you don't tell the brain that you're having a defecation reflex, you're going to be in trouble. So the brain will then send down a signal and determine what this external sphincter is going to be doing. All right, so if we want it to stay closed, we're going to stimulate this skeletal muscle and keep that anus closed. If we want defecation to proceed, we can relax the external anal sphincter and defecation will proceed. All right, so that is the nervous system control of defecation. Finally, a few of the disorders. There's a lot of disorders with the digestive system. These are just some of them. Ulcers. Ulcers are basically lesions in the lining of the stomach. The acid will get in there and you can cause pain. There's a major cause that they identified years ago, a bacterium. And the bacterium is called Helicobacter pylori. Interesting study in that the person who, identified this, his name is Barry Marshall. To prove this, he actually drank cultures of the bacteria and gave himself ulcers. And that kind of proved this connection between a bacterium and ulcers. Before this, most people thought about, ulcers being, stress related. Now, stress is a component of it, but they also found the additional bacterium that can cause it. NSAIDs can cause it. NSAIDs will thin the mucus layer. And if you thin the mucus layer, that stomach gets more susceptible to the hydrochloric acid. All right, so typically when someone has an ulcer, You want to treat the acid, so you reduce the acid levels, so maybe give a proton pump inhibitor and then give an antibiotic to kill the bacteria. Diverticular disease, these are outpouchings that happen in the wall of the colon. So the wall can get a little weak and get pushed out. This can cause inflammation. This can also trap feces in the little outpouchings, and then they can get infected. Cancer, obviously, is a big issue. It's one of the top deadliest cancers in the human body, behind lung cancer and breast cancer. There's a big genetic component there. Diet can increase risk of colon cancer, as well as alcohol. And then there's hepatitis. Hepatitis is due to it's due to viruses or drugs like alcohol that damage the liver. The viruses, there's a number of different viruses that cause hepatitis. There's hepatitis A, which is typically picked up with contaminated food or contaminated water. And then there's hepatitis B and C, which are usually contracted from blood, so either dirty needles, or it can be contracted by other fluids that contain the virus. But that's a little more of a microbiology topic. Okay, so that ends the digestive system. We're going to kind of continue the digestive system in the next chapter when we're looking at a little bit of nutrition and metabolism, but that's going to be condensed quite a bit. So work on this digestive system, work hard on it. Let me know if there's any specific questions, and good luck.